Today, I'm going to be talking about my part in Project 11 Plus, which I've called the Black Soldier Flies Army Microbial Responses to Changes in Diet Composition. Um, so this is the part three in a three-part seminar series where in October, Aaron and Sienna presented on the impact of macronutrients on phenotypic plasticity of industrial insects. In November, Michael Smith did a presentation on a transcriptomic analysis of the impact of diet composition on the BSF gene expression in the gut. And then this month, I am going to be talking about the little guys, the microbes that are actually inside of black soldier fly and surround the black soldier fly in their frass. And so we're going to look at their changes in response to dietary composition. And so this is a big project made up of a lot of different people working at the three research institutions in the center. And so it includes Dr. Beamer and Dr. Tomberlin from Texas A&M, Drs. Hoffman, Meyer, and Jordan from Mississippi State, and Dr. Picard from Indiana University at Indianapolis. And then we've got a cohort of students and other researchers working on this from MSU, Texas A&M, and IUI, along with our two IAB mentors, Chris Warburton from Intosystem and Pat Crowley from Chapel Farms. So the goal of Project 11 Plus is to determine the impact of macronutrient content on life history traits, genetics, and microbiome of the black soldier fly, mealworm, and cricket, the big three of industrial insects. And so this is a pretty ambitious project. It's multifaceted. So we have data like survival and growth that are that indicate the life history of these insects grown on these different diets. We also have gene expression data of each insect, and we've also surveyed the community and function of the microbiome within these insects. And so the different projects that are included in Project 11 Plus is the BSF lab scale, which is just a small scale study looking at three different macronutrient ratios and how the BSF respond to this on a small scale. And then there's a benchtop scale, which is quite a few more black soldier flies involved and about the size of a shoebox. One macronutrient ratio was included in this study, and it was determined from the lab scale study. And then there's also a microbial inoculation component in that study. And there's also a mealworm study that deals with three macronutrient ratios in a choice assay, and a cricket study that deals with the same thing. So today I'll mainly be talking about the BSF work because we don't have sequencing data back quite yet from the mealworm and cricket, but I do have some new results to share on the black soldier fly benchtop study. So first, I'm going to talk about the lab scale study in black soldier fly. This is, as I mentioned earlier, measuring responses on different dietary treatments. And so the macronutrient profile was manipulated on these different synthetic artificial diets. And there's a carb bias diet with a 1 to 5 protein to carbohydrate ratio. There's a balanced diet with a 1 to 1 protein to carbohydrate ratio diet, and a protein bias diet, which is a 5 to 1 ratio diet. There was also a Gainesville control diet, but we won't really be getting into that today because it's hard to compare Gainesville to the nicely controlled artificial diets. And then these were grown up on a small scale in these 50 milliliter conical tubes. We measured the life history, the gene expression, and the microbial community and expression after this. And so I'm not going to go into too much detail about the artificial diet formulation, but basically the way I understand it is that we have these diets that can easily be manipulated with regards to their macronutrient ratio, and that would be carbohydrate and protein. And this is the rest of the diet is for those who are interested, but I won't get into that too much. And then the way that this experiment was set up was that 7-day-old neonates were weighed in groups of 10 and placed in those conical tubes with 10 larvae per tube. The tubes were monitored daily to look for prepupation levels. And then when the tube displayed 40% prepupation, it was harvested. And then that's when the life history traits were measured for larvae. So the growth and survivorship were determined. And then I was sent samples to dissect for microbial analysis and gene expression analysis. Okay, and here's a quick recap of the results that Aaron and Sienna presented a couple months ago. So they found in the lab scale study that the balanced diet, so that 1-to-1 protein to carbohydrate ratio diet, resulted in a significantly higher larval weight, with the protein biased diet and the carbohydrate biased diet having lower weights. And then as far as survivorship goes, the protein biased diet, or the 5-to-1 diet, had lower survivorship overall of the black soldier fly larvae. And then when you're looking at the days to pupation, the carb biased diet took significantly more time than the other two diets to reach pupation. And so what we're faced with are these different responses to dietary composition. You see trade-offs in this data, and but overall, it seems like the balanced diet was the most optimal diet in this scenario. And then here's a quick recap of the gene expression results. And so I'm going to go a little bit into what this means. This is from Michael's presentation last month. But what we can do is we can measure the expression levels of all the genes in black soldier fly, and then figure out which ones are differentially expressed, are dysregulated in a specific diet treatment. And then if you look at the box plots on the left, the diet treatments go from the carb biased diet as the 1-to-5 diet, the balanced diet in the middle is the 1-to-1 diet, and the protein biased diet on the right as the protein biased diet. And then you can group these transcripts by their expression patterns. And so, for example, in group A, which I'm not sure if you can see my mouse cursor, but in group A, these 165 transcripts that are differentially expressed, or sorry, that follow the same expression pattern, are downregulated in the carbohydrate biased diet, upregulated in the balanced diet, and downregulated in the protein biased diet. And then the opposite is true for group B, where they're upregulated in the carb biased diet, downregulated in the balanced diet, and upregulated in the protein biased diet. And then on the right is a characterization of the functional terms that are enriched inside of these differentially expressed transcripts. And so taken together, the life history data, or results in gene expression results, kind of underscores black soldier flies' resilience to these changes in diet composition. And so, for example, there were a few other examples that Michael brought up in his talk. But if we look at group A, the genes with high or upregulation in the balanced diet, we see that it kind of mirrors the weight data from the larvae, from the phenotypic or life history data that we had. And we can see that 48 of the transcripts that are upregulated in that balanced diet were involved in developmental regulation, which could explain why in the balanced diet for the life history data, they had higher weights. And the same is true with the 5-to-1 and 1-to-5 diet. Those genes involved in developmental regulation were downregulated in those diets, resulting in which correspond with the lower weight in the carb biased and protein biased diet. And so at my job, hopefully, what I will do today is tie it all together and share what role the microbes might play in black soldier flies' resilience. And so with that, I'll start my part in the BSF lab scale study. So a quick methods overview. Once again, we subjected larvae to these different dietary compositions. We left out the Gainesville control for this study because, once again, it's hard to compare Gainesville to these nicely controlled artificial diets. And so we took 9 whole larvae guts and 9 subsamples of frass from these different dietary enclosures, and plus a diet without larvae that was grown alongside to the experimental samples. And we sequenced the total RNA, or the transcriptome, of these samples. And so a quick recap on why we used transcriptomics for this is because transcriptomics can comprehensively detect the active microbes and insects. So if we go back to our basic molecular biology, DNA is transcribed into RNA, and this process only happens whenever an organism is alive and active. And so we're getting rid of any, dormant bacteria or even dead microbes that are inside of the black soldier fly by using RNA or transcriptomic data, so that we are really only measuring the levels of what microbes are active in there. And so quick overview is you take your industrial insect of interest, you sequence the total RNA, and then what I do is align the reads that you get from sequencing to the host genome and get rid of those because I'm more interested in the microbes than the actual host. And those reads that align to the host go to Michael, and he does the gene expression analysis of the host. And then you can take the unmapped reads and assemble them based off overlapping regions within the reads. So you have transcripts and viral genomes in that fraction of the data. And you can also assign taxonomy to the remaining reads, so you can predict which bacteria or fungus or virus that a specific read comes from. And then you can measure the abundance of those microbes by how many reads are assigned to that particular taxa. And some applications that are involved with this is with the assembled reads. You can do all kinds of stuff. You can detect known and novel virus genomes. You can predict the function of microbial transcripts based off of their sequence. You can also determine expression levels of these microbial transcripts to determine maybe which ones are important in response to changes, for example, diet composition that we used here. And then in the unassembled reads, you can do a microbial community analysis. And a big perk to this method is that taxonomic detection is not limited by amplifying some kind of like barcode gene, such as the 16S gene, which you'll only get bacterial results if you use just a 16S based study, or ITS gene, which you'll only get fungal results. So we're simultaneously able to detect viruses, bacteria, and fungi all in one assay. And then so the methods, so what I had to do to get the data to be able to be processed is I was sent the larvae, I dissected the guts. We had 9 larvae in total from 3 random tubes. I sterilized the cuticle of the larvae beforehand in ethanol, then dipped it in water to clean it off, and dissected the guts in RNA later, which is a solution that keeps the integrity of the RNA in place. And then we used RNazole for RNA isolation. And then concordantly, we took a gram of frass from each enclosure and extracted the RNA from that too. So RNA was isolated, the purity, concentration, and integrity was assessed. And then we prepared the libraries for sequencing. We didn't do ribosomal RNA depletion or mRNA enrichment so that we could keep all host and microbial reads. And then we sequenced on an Illumina sequencing platform. And then, so I run it through this computational pipeline that I kind of Frankenstein put together. And what you do is you take the resulting reads that you get from sequencing, you inspect them and get rid of the low quality reads. You map them to the insect genome and discard those mapped reads or give them to Michael to do the insect gene expression analysis. And then you can assign taxonomy in abundance using Kraken 2 and Bracken, which are just softwares that are meant to do that, and then do a microbial community analysis with those reads. And then you can assemble the rest of the reads. We cluster each transcript based on sequence similarity so that you have one representative transcript per redundant transcripts as your representative of the function of that transcript. We used a 90% sequence similarity, and then you can annotate those transcripts using blast-based analyses where you can figure out maybe what taxa or viruses are in there. And then you can also use another tool that annotates the function of these transcripts, and then you can do your analyses. And so here are the lab scale results. So first, we noticed that diet composition affects diversity of the black soldier fly gut and frass microbial community. And so this was measured as alpha diversity. And so on the left, you can see alpha diversity of the larval samples. And on the right in panel B, you can see alpha diversity of the frass. And immediately, what we see is that in that 1 to 5, so the carbohydrate-rich diet, in the larvae, the diversity is going up. And so when I say diversity, I'm talking about the number of species, so the richness of taxa in a sample, and also the evenness of the distribution of the taxa in a sample. And so we can see that in that carbohydrate diet, at least in the larval gut, the diversity goes up compared to the balanced diet in green and the protein biased diet in blue. So this is suggesting, and it kind of makes sense, that the microbes are using the carbohydrates in the diet as a source and are proliferating in the side of the larvae gut. And then the opposite effect is kind of seen in the frass, where we see actually lower diversity in the carbohydrate-rich diet compared to the balanced diet and the protein-rich diet. And so next we measure beta diversity, which is, you can think of it as the overall community within each sample, and then you can compare them by reducing the dimensions of your data set and putting it onto 2 dimensions. And you can basically see which samples are more similar to each other by their clustering patterns. And so once again, we have the larvae on the left and the frass on the right. And so looking at the larvae, we see these pretty nice clusters of corresponding with each diet. You can see a shift in the carbohydrate diet to the right side of the x-axis, while the balanced diet and the protein biased diet in green and blue are kind of clustering together, which is similar to what we saw in the alpha diversity results. And this was a significant result. You can measure this if these diet compositions are affecting the overall community statistically using a permutational ANOVA, and we did get a significant result on that. And then looking at the frass, we see this very distinct difference between the carbohydrate diet, carbohydrate-rich diet, and the other 2 diets, where you see the yellow dots all the way to the right on the x-axis, and then the other 2 diets kind of cluster together on the left side. And it's separated by 85% of the variation is captured in that x-axis, and so, or the variance, sorry, is captured in that x-axis. And so these microbial, the composition of the microbial community are very different between the carbohydrate diet and the other 2 diets. And this is, once again, the same result that we saw with regards to alpha diversity. Okay, so next is a snapshot of the most abundant taxa in these 2 different samples, the larvae, or sorry, in the diet compositions of the larvae and the frass. And so what we see right off the bat in the larvae is all of that yellow, which corresponds to a fungal taxa. And this is pretty interesting. Until recently, it seems that the role of fungi in the black soldier fly microbiome has been kind of ignored. But here we see basically dominance of a fungal taxa in the larvae gut microbiome. We also see this other fungus, this Pichia fungus, and What's interesting about both of these taxa is that they are known in cactophilic Drosophila, so flies that like to lay their eggs in rotting cactus flesh, make associations with species of both of these genera in the wild. And what they do is they lay their eggs in the rotting flesh of the cactus. And the rotting flesh of the cactus creates these toxic fatty acids that these fungi are able to degrade, and the larvae of the Drosophila can stay alive. And so when I started thinking about rotting cactus, I immediately started to think of what substrate black soldier fly kind of gravitate towards in the wild, and it's these compost and decaying matter and things such as that. And so these fungi may play some role in detoxifying their meals or breaking down some of the toxic substances inside of the substrate. So I found that pretty interesting. And then when we look over at the frass, we see that in the carbohydrate bias diet, that purple is a Klebsiella species, and it basically dominates the microbial community there, which we also see that purple Klebsiella in the larvae at pretty high levels too. But in the protein bias diet and the balanced diet, there's much less of that Klebsiella genus, and Lactobacillus and Streptococcus kind of take over in that greenish color and the pinkish color. And like I said, this is a snapshot of the most abundant taxa. Using the method that we use, the transcriptomic method, it returns a lot of taxa. Most of them are low level though. From measuring what taxa have greater than 1% average makeup in the relative abundance, it's just 12 taxa in the larvae and 14 in the frass. And so using the top 10 from each the frass and the larvae is a pretty comprehensive look at the dominant taxa in their microbiomes. So like I mentioned earlier, we also had some control diets that were run alongside the experimental samples. And so this is a snapshot of the relative abundance of the microbes inside of those samples. And so once again, going from left to right, we have the carbohydrate bias diet, the balanced diet, and the protein bias diet. And immediately what we see is another fungal species that is highly represented in the carbohydrate bias diet. So that orange color in the 1 to 5 diet is mucor, which is a fairly common fungus. I think it's the fungus that is like in bread mold and things like that. But we don't see that represented in the balanced diet or the protein bias diet. We see basically Klebsiella is dominant in those diets. And so what's pretty interesting is that, and here are the taxa that were also top taxa in the experimental samples. So we see that some of these bacteria were present in the diet before we put the larvae in there. But what we do see is that larvae has a pretty big effect on the frass microbiome. And I'm sorry these aren't the same colors in both of the charts. I should have done that. But what we see is that none of that mucor species is represented in the 1 to 5 diet for the frass and the experimental samples. In fact, the Klebsiella genus that's highly represented in the 1 to 5 diet is highly represented in the 1 to 1 and 5 to 1 control samples, which is not true with the 1 to 1 and 5 to 1. The Klebsiella is not the dominant species. And so basically what this is showing is that the larvae are affecting their community around them, and the microbiome of the frass certainly changes with the addition of larvae. Mm. And so some takeaways from the community analysis is that microbiota respond to changes in diet composition. We saw that in the diversity analyses. We also see that fungi might play an important role in nutrient metabolism from larvae. We saw that in the relative abundance of that fungal species throughout the larval guts. And then also, we saw that larvae impact frass microbial composition. And so next, we know who's there, but do we know what they are doing? And a nice thing about this transcriptomic data is that you can infer what functions are represented inside of this microbiome using those assembled transcripts that I mentioned earlier. And so I'm going to go into more detail in just a second. I know this is a lot to look at right now, but I just kind of wanted to go over how to read this plot. It's very similar to what I showed earlier for Michael's results. So what we did is we found what microbial transcripts were differentially expressed in each diet, treatment, and clustered them based off of similarity in expression patterns. So once again, going from left to right, you see the carbohydrate biased diet, the balanced diet, and the protein biased diet for the box plots. And these differentially expressed transcripts were clustered into three groups. There were right under 500 differentially expressed transcripts. And the main expression patterns that were assigned were upregulation. So in group A, upregulation in the carbohydrate biased diet and downregulation in the other two diets. Group B was really no dysregulation in the carbohydrate biased diet, downregulation in the balanced diet, and upregulation in the protein biased diet. And in group C, we saw downregulation in the carbohydrate biased diet and upregulation in the balanced and protein biased diet. And so on the right, once again, are the transcripts that are making up functional enrichment terms, which are things like gene ontology terms and pathways that are being detected more than you would expect at random. And so zooming in on group A, which is the majority of the differentially expressed transcripts, and the pattern that this is following is upregulated in the carb biased diet and downregulated in the balanced and protein biased diet. And what we're seeing is that the function of the microbes is reflective of the diet composition, which makes sense. So it's like you're sowing the seeds for these microbes to have functions like carbohydrate metabolism, carbohydrate transport, and cellular respiration, which is a cellular process that metabolizes carbohydrates into energy for cellular organisms. And so what we're seeing is these functions involved in carbohydrate metabolism are upregulated in the carbohydrate biased diet. There are also other metabolic pathways that are present here. We have nucleotide metabolism, which could be part of the pentose phosphate pathway, which is also part of cellular respiration, and protein metabolism. And so it's pretty interesting that basically what you would expect, the functions you would expect to see in the upregulated transcripts in the carbohydrate biased diet of the microbes is what we are seeing. And then also in the other two groups of transcripts, we see the genes that are upregulated in the protein biased diet. There are four transcripts that are four transcripts that are involved in protein metabolism. And then in group C, we see transcripts like protein metabolism upregulated in the balanced and protein biased diet, a cellular respiration gene, and some other just nucleotide metabolism and translation genes that are upregulated. And there are the two categories in gray, which are either other or no annotation. Other means some genes that were not associated with an enriched functional term, and the no annotation means that the transcript was too diverged to infer its function. And so another takeaway from the functional analysis or the metatranscriptomic analysis of the differentially expressed transcripts in the bacteria is that it's a multi-domain effort. And so what I've done here is taken all of the differentially expressed transcripts and mapped them to their most specific taxonomic annotation. Basically, what you need to look at is the blue versus the green, or the bacteria versus the eukaryotes. And we can pretty much assume that all of the eukaryotes are fungi because there should be no more insect transcripts or reads that were present in the analysis. And we can see that there's quite a few bacterial transcripts, specifically those of gamma proteobacteria, that are differentially expressed in our data set. But eukaryota is also making up a substantial fraction of the differentially expressed transcripts, suggesting that the breakdown of the nutrients are macronutrients inside of the different dietary compositions are not limited to bacteria, but also fungi. And so some takeaways, overall takeaways from the functional analysis is that one, microbial function is reflective of diet composition, which is expected, and it's nice to see that in your data. But another thing that I kind of was interested in while I was looking at this data is that microbes may be hogging the nutrients in the 1 to 5 diet. And bear with me. So we saw increased diversity in the microbes of the larval gut. Okay, so there's more species present, is what that means. And we saw a decreased weight in the carbohydrate diet. You can see that in the top left. And we had an increased time to pupation in the carbohydrate biased diet in the top right. And so this could be suggesting that the microbes are utilizing the nutrients before the insect is able to use them. And it could be because there's an increased diversity of microbes within the gut, and especially those fungal taxa that are doing those basic cellular processes like respiration, where they're turning the carbohydrates into ATP that are not usable for the larvae. Another line of evidence that suggests this is that lysozymes are upregulated in the gene expression data that Michael presented last month. And lysozymes are antimicrobial enzymes that are known to play functions in immunity and things like that, to where they will actually break down bacteria. And so this all points to that these microbes may be hogging the nutrients, and then the black soldier fly may be using the microbes as a nutrient source after they, as the diversity of the microbes are going up inside of the gut. And then finally, the functional analysis tells us that this is a multi-domain effort. Okay, so next is some new results from the BSF benchtop study. We're kind of switching gears now to a new analysis that I mentioned earlier. So what happened is we took a shoebox size enclosure for the larvae in black soldier fly. We used the 1-to-1 artificial diet, so the balanced diet that seemed like it was the best or the optimal diet of the 3 artificial diets that we fed in the lab scale study. And so we put 1,000 larvae on this diet. And then in some samples, we inoculated them with a Klebsiella species, which was actually taken from the gut of black soldier fly. And as we saw, those Klebsiella species are definitely present in the black soldier fly gut. And some of these shoeboxes were inoculated with Klebsiella, while some were not. And then we had 3 shoebox replicates per treatment. So 3 Klebsiella inoculated and 3 non-Klebsiella inoculated treatments, and then 6 shoeboxes total. And then we used the same sequencing methods as before, except for in library prep, we actually enriched for microbes to study the microbiome. And what that does is it just gets rid of some of the host reads so that we have more microbial reads to work with. And so here's a recap on what Aaron and Sienna presented a couple months ago on the weight and survivorship of the black soldier fly after inoculation of Klebsiella, which WO stands for without Klebsiella and W stands for with Klebsiella. And interestingly, we did not see any difference in the weight or survivorship statistically between the Klebsiella inoculated black soldier fly and the non-Klebsiella inoculated black soldier fly. And so while we didn't detect this statistically, we did see that there was slightly increased weight in the BSF that were inoculated with Klebsiella, and also a slightly increased survivorship in the black soldier fly that were inoculated with Klebsiella. And these could have implications biologically. If you scaled it up, we would possibly be able to detect these statistically, but with the data that we have, we did not actually detect statistical differences. And so now I will go into the community analysis for the BSF benchtop. And we did not observe, similar to the life history traits, we did not observe any changes in alpha diversity statistically for the larvae or the frass. What we do see is a slightly decreased diversity in the bacterial or the Klebsiella supplemented larvae, but we don't really see that difference in the frass. So this is kind of agreeing with what we saw in the life history data. We also didn't see any significant differences in beta diversity between the larvae or the frass. We do see some semblance of clustering in the both the larvae and the frass data, but either we didn't have quite enough replicates to detect statistical differences, or there's just too much variation in the data set to detect statistical differences. Okay, and so here are the top taxa that were represented in the benchtop study. So right off the bat, what I was interested in was to see if that fungal species would still be represented in the larvae, but we do not see that in the benchtop study. We don't see very much fungal species represented at all in the top taxa. That Talaromyces, the gray species in the gray at the bottom of the legend, is a fungal species, and it's present in the non-microbial inoculated larvae. So the yes and the no corresponds to if they have Klebsiella or not. But we do see some species that were represented in the previous data set in the lab scale, including Klebsiella, which is what we inoculated with. So I'm glad that we actually detected Klebsiella in there, although they were present beforehand, and Lactobacillus is present in there. Staphylococcus and Streptococcus species are both main constituents of both the larvae and the frass microbiome. And similar to the previous data, just over a dozen taxa make up at least 1% of the relative abundance of both the larvae and the frass. So this is a pretty comprehensive look at the microbiome, especially the dominant microbes. And so I also wanted to see if there was an effect of, or if we could detect the effect of Klebsiella supplementation. And similar to the life history data, we didn't detect any significant differences between samples with Klebsiella supplementation versus samples without Klebsiella supplementation in both the larvae and the frass. And this could be because using the transcriptomic method, we cannot really detect species level assignments, and we may not should trust species level assignments from the transcriptomic data, but nonetheless, we didn't see any difference at the genus level. And this could be because the Klebsiella species makeup may have changed at the species level, but maybe not at the genus level. So I'm working on a way to see if we can have species level estimates to see if the actual species of Klebsiella that we inoculated with has changes from the bacterial supplement samples versus the non-supplemented samples. Okay, and here's a look at the functions that are enriched in the BSF benchtop study. Okay, so this is going back to Michael's presentation from last week. He saw that there were moderate clustering So moderate differences between the Klebsiella supplemented group and the non-Klebsiella supplemented group in the black soldier fly gut. So this is not the microbe data, this is actual black soldier fly data. And there was actually a pretty small amount of differentially expressed transcripts in the black soldier fly data, which also corresponds with what Aaron saw and Sienna saw in the life history data. My principal component analysis looks very similar to Michael's. There is more variance on the x-axis, showing that there's more separation between the Klebsiella inoculated samples and the samples without Klebsiella. Basically, what this is telling us is that the effects of the treatment were actually detected. There are much more differentially expressed genes than what Michael saw in the microbes of the there's much more differentially expressed genes in the microbes than there were in the black soldier fly. And so this plot right here is a volcano plot, and the way you read that is going up the y-axis is the significance value associated with differential expression. So these are genes that are actually differentially expressed, and then going along the x-axis into the positives numbers are the Klebsiella, are the genes that are upregulated in the Klebsiella supplemented groups, and then going to the left in the negatives in that pink color are the genes that were downregulated in response to Klebsiella supplementation. There was a total of 463 genes that were differentially expressed. 244 of those were upregulated, and 219 were downregulated. And so now what I want to do is see what functions were enriched in this data set. And this chart on this slide is a way to assign a parent functional term to enriched functional terms in your data set. And so what you do is you have all of these functions associated with differentially expressed genes, and you want to see if those functions are represented more than you would expect to be there at random. And so you get a lot of functional annotations associated with this, and some of them may be functional annotations that are basically telling you the same thing over and over again. And what this does is it clusters similar functional terms all together and has one single parent term to represent all of the functional terms that are enriched. And what we see is that in the overall and the differentially expressed genes of the benchtop study, there is functions like oxidative phosphorylation, nucleoside metabolic processes, and proton transmembrane transport, which once again, we're seeing more basic cellular processes involved in cellular respiration enriched in these differentially expressed gene sets. And so when we look at just the upregulated transcripts, so just the ones that were in purple from that plot earlier, we see functions like response to cold, GMP metabolic process, things involved in DNA replication and translation. So really not much to glean there. But when we just look at the downregulated transcripts, we see those respiration terms again, like oxidative phosphorylation, ATP metabolic process, proton transmembrane transport, like the electron transport chain, going back to basic biochemistry. And we see that all of these terms are downregulated. And so I was kind of interested as to why that is. Why are these functions being downregulated in response to that Klebsiella treatment? And so I started looking at what taxa are differentially expressed. And what we see is that fungi make up a relatively small part of the taxa that are differentially expressed. But if you look at the downregulated fungal samples, it's basically all of them. So out of the 29 differentially expressed fungal transcripts, 28 of them were downregulated. And so then I wanted to see, are these fungal transcripts the ones that are the basic cellular processes functions that are enriched in the differentially expressed genes? And basically, we found that is exactly what's happening, is that fungi are accounting for all of those cellular respiration terms that are enriched in the downregulated genes and the overall differentially expressed genes functional enrichment. And so this is pretty interesting. And so this could suggest that Klebsiella is competing with the fungus, and which is in turn down regulating those cellular processes like cellular respiration and pathways associated with that. In the bacterial functional enrichment, so these are the rest of the transcripts that are associated with bacteria. There's not much to glean yet, but as you can see, those are the rest of the functional terms that are enriched in the overall functional enrichment of the differentially expressed transcripts. And so the overall conclusions on this is that, similar to what we saw in Michael and Aaron's in Sienna's presentation, that Klebsiella supplementation did not really have a huge effect with regards to community, but we can detect changes at the microbial expression level. And something that is interesting that's in line with the down rate, or the lower diversity of, so you can see on the right, the lower diversity of the Klebsiella supplementation. So we saw that there's down regulation of these fungal cellular processes, and it could be because the Klebsiella is driving away these fungi, which is also lowering the overall diversity of the sample. And this may be through something like competition, but although we couldn't detect this statistically, it could have biological relevance. And so overall, our conclusions for Project 11 Plus is that we can get these multi-tiered results that are illuminating some of the complexity behind black soldier flies resilience. So we're seeing that diet is affecting BSF gene expression and the microbiome, and the gene expression of the black soldier fly is affecting the gene expression of the microbiome. The microbes are affecting the gene expression of each other, which is all informing what we're seeing in the life history data. And so it's a big and complex data set, but it is fairly interesting whenever we start making strokes at the big picture as to what's going on. And there's more on the way. Some of the cricket and mealworm samples were sequenced after an unfortunate blip in the road with shipping and some samples melting, but they redid library prep with low input enrichment, and these sequences actually passed QC. And so the rest of the sequences the rest of the samples are being sequenced right now, and we're waiting for that data. And also the lab scale BSF manuscript is in prep.